you're ready to go, and we're all, I think, take it away.
Right. All right. Good afternoon, good morning, good evening, everyone. My name is Sabieh Anwar, and it's a great honor for me to be talking in this workshop. Can you all hear me properly?
Yes, yes.
All right.
Yes.
Okay.
Yeah.
So my talk is going to be an exploratory talk. I would like to explore a new area that I'm learning these days. I'm a physicist and I generally love quantum physics. And this is a workshop on biophysics. So I've always been excited and enthused by the possible marriage between the two areas. When I was a PhD student, I heard a talk by Anton Zeilinger, the famous quantum physicist, and he said that people try to connect consciousness with quantum physics, and they do so because neither do they understand quantum physics nor do they understand consciousness. So they think there must be a wedlock between them, some interrelationship between them. But it turns out that quantum mechanics is, over the years and over the decades, have been talking to biology in a more intimate fashion. So I would just like to give an overview of what excites me these days and some of the work that we're trying to do uh, with some of our students. So I would like to share my screen. All right, so I'd like to talk a little bit about the possibility of interconnecting quantum mechanics with biology. Quantum mechanics, as we all know, was discovered or invented about 120 years ago by Planck, and then it was consolidated by Einstein, Bohr, Schrödinger, Heisenberg, and the likes of these prodigies. But generally, we live in a classical world. What this means is that the temperatures that we work with and the kind of objects that we deal with, they are classical. This means that our temperatures are roughly around room temperature, and we generally deal with large systems, macroscopic systems. Quantum mechanics, however, it becomes more manifest in really cold environments and at the really small scale. So if you would like to cool this universe to somewhere around 1 Kelvin, minus 272 degrees centigrade, and you would break it apart into pieces, and you have microscopes and the means to look at individual particles, individual entities, you would rather enter the quantum realm. So the classical realm is really average quantum mechanics. And if you would really like to look at quantum mechanics in its full glory, you have to get away from this ensemble averaging. You have to look at the tiny, and you have to look at the cold. So that's why the relationship between biology, which is a classical system, we deal with large objects and hot, warm objects, dirty objects, wet objects in biology. And that's why the relationship between the two has defied many attempts of reconciliation. However, recently in the past 2 or 3 decades, there has been a growing interest in looking at the connections between quantum mechanics and biology. And I will just highlight a few examples today. So, by the way, just to give you an example of quantum technologies in its full glory. So right now we are seeing the heydays of quantum mechanics, in the sense that our quantum technologies have matured to such an extent that we can build quantum computers, which have more than 50, 60, 70 qubits. That is, they break the quantum ascendancy critical threshold, and they can do computations that are really useful and that can be that can be achieved in a fraction of a second, something that would have taken a normal supercomputer or a conventional computer thousands of years. But these quantum computers are not in any way similar to the desktops which we use, the laptop that I'm using right now for my presentation. These are strange, big, bulky devices that are kept at liquid helium temperature, really cold, around 1 Kelvin or the whereabouts of Kelvin, and pulses go uh, into these qubits and into these superconducting circuits, and then the information is read out, and everything has to be completed in the fraction of a millisecond. Uh, and so you have to keep this system really cold, because interaction with the environment, which is called decoherence, really breaks away the coherence, and the quantum mechanical properties, they just fizzle out. The cat is either found alive or dead. And that's what we see in biological systems. The environment is so pervasive and it's so overwhelming that it's very hard to see quantum mechanical effects. So how does quantum mechanics have a role to play in biological systems? Well, I would look at three aspects of quantum physics which are important for biological processes, or at least they are perceived to be important for biological processes. By the way, this is a new area. This maybe about 20 years, the field of quantum biology is blossoming these days, but it's a new field. We are still in the neonatal phases. About 20 years ago were the first few papers on quantum biology came out, notwithstanding Erwin Schrödinger's book in 1923 about what is life, but that is not tacitly quantum mechanical. So the first aspect that I would like to highlight is quantum mechanical tunneling. Now, tunneling is a phenomenon that is routinely seen in the laboratory, and in many biological and chemical processes, tunneling is at work, especially at the molecular scale. On the screen here, you would see on the top left a scanning tunneling microscope image of individual cobalt atoms. And on the bottom left and the right, you will see that with the help of this machine called the scanning tunneling microscope, you can address and negotiate with single atoms, pick them up, and put them at locations of your desired desire on a substrate, and you can construct different kinds of quantum corals and other kinds of geometrical structures. So with the help of tunneling, you could not only address or image atoms, you could also manipulate them. So this is the pinnacle of nano manipulation. And how does a tunneling microscope work, and what is quantum mechanical tunneling? In quantum mechanical tunneling, you have a metal tip, maybe of tungsten, and you have a surface, a conducting surface, and you hover this metal tip over and above the surface, keeping, and then you expect that electrons will hop from the conducting sample to the metal tip or vice versa, and they will hop over an insulating layer, even though electrons do not appear to have sufficient energy to cross this barrier. And they do so because of quantum mechanical tunneling. So the idea of quantum mechanical tunneling is that a particle can go where it is classically forbidden to do so. It can cross a barrier which is classically insurmountable. So this is the essence of quantum mechanical tunneling. And tunneling, by the way, is an extremely important natural phenomenon. So on the top left, you can see a cartoonic image of quantum mechanical tunneling. So a person would like to climb a hill and go to the other side. Classically, the only possibility is that this person musters the required energy, climbs the hill, and then descends on the other side. Quantum mechanical tunneling tells us that the person, even with an energy that does not cross the barrier, the threshold barrier, could still move from one side to the other, in some ghost-like fashion, in a sense, and appear on the other side. And the probability of this process happening is larger if the energy of this person is higher, or the barrier is, the width of the barrier and the height of the barrier is small. We see exactly the same process in natural radioactivity, in which an unstable nucleus harbors a helium nucleus, two protons and two neutrons. And these protons and neutrons, which form an alpha particle, can tunnel out of the nuclear regime and come outside, and they become radioactive particles. They come out as radioactive particles. So they have the ability to cross this barrier. Now, if you look at the various radioactive nuclei, polonium, radon, radium, and uranium, you will find out that the particles that are coming out from this nucleus, mostly they have roughly similar, same order of magnitude energies, in the order of less than 10 mega electron volt. However, the lifetime of these nuclei straddles a very large time scale, right from a nanosecond to 6 billion years, which is half the purported life of the universe, even though all the energies are the same. So this also gives us the idea that the properties of radioactivity acutely depend upon the parameters. You change the energy by a slight bit, the rate of the reaction might change. Now let's enter the molecular world. In the molecular world, we are very familiar with the geometry, the umbrella-like structure of the ammonia molecule. So the ammonia molecule comes in this pyramidal structure, and we know that it can invert into its to the other side of the umbrella. And if you look at the energy level diagram for this conformation, you have two cusps, and those cusps have the same energy, but they're separated by a barrier. Now, classically, this would require a large temperature to surpass this barrier, but even at low temperatures would such an inversion occur, and that is because of quantum mechanical tunneling. So tunneling describes the inversion of molecules. Now enter the biological world. I'm not a biologist, so I really don't know much biology, but how quantum mechanics enters the picture is something that is really interesting. Now, a catalyst or a bioenzyme would have a certain, there's going to be a barrier between the reactant and the product phases. And the purpose of a catalyst or an enzyme, as we all know, is to lower this activation barrier. Now, at room temperatures, most reactions in the presence of a catalyst will have enough thermal energy to surmount this barrier, and the system can go from the reactant to the product phase. But if we lower this temperature, we are going to suppress the thermal effects. And in such a case, the propensity or the probability for this reaction occurring is going to drop exponentially because of the underlying mathematical structure of how enzymes work. Now, it appears that the process, even though becomes classically insurmountable, quantum mechanical tunneling can come to the rescue and allow this process to proceed through a tunneling pathway. Just last year, a paper came out in the Angevante in which a molecule, nitrobenzaldehyde, ortho-nitrobenzaldehyde, would, in normal circumstances, at room temperature, would convert into this molecule, number 2. However, and when the temperature is lowered, the reaction is not favored because the activation barriers remain too high for the reactant phase to go into the product phase. However, a new kind of tunneling product is seen, which can only be described by quantum mechanical tunneling. So if you look at the bottom right figure, the from the phase number 3 to phase number 4 through an intermediate ketene molecule, and by surpassing a potential barrier, would favors the formation of this product, which is only describable by quantum mechanical tunneling. So this has huge implications, and this is just the tip of an iceberg, I think. We know that electron transfer and proton transfer reactions are really important for many biological enzymatic processes, and most of biology happens at room temperatures. But for processes in which, suppose we have large protein molecules, and we would like to see whether a proton or an electron can tunnel from one end of the molecule to the other, which is classically really difficult, even at room temperatures. Could tunneling play an important role here? So this is an important point of investigation currently in quantum biology circles. Lithium-ion batteries. Lithium is another small atom, and the possibility that lithium can tunnel through a membrane in lithium-ion batteries also raises the possibility of tunneling activated batteries. And are there catalysts that can exclusively work with tunneling? So all of these are currently open questions, and some areas to look out for in the field of quantum biology. In fact, I was looking at the literature while I was preparing this talk. It is also possible to directly image these tunneling electrons with the help of nanoplasmonic gold nanoparticle antennas, which can radiate out, and you could see an image through electron energy loss spectroscopy. So this is not something which is just a matter of fiction. Recent work has actually taken an upshot, and is really we see this research happening at an accelerated pace. The second aspect of quantum mechanics, which has a possibility of becoming important for biological systems, is what is a particle itself. So quantum mechanics has this strange property that in some cases particles, these particles could be light, which is important for photosynthesis. These particles could be electrons. Most of the biochemical processes are electron transfers or proton transfers. So whatever they are, these particles can have a dual character. They can act like particles, or they can act like waves. And the simple cartoonic description of how we distinguish a particle from a wave is that if you put a barrier in the path of particles, the particles are blocked by the barrier and they cannot bend around the barrier. However, for waves, they can easily spread out across barriers and reach hitherto or otherwise forbidden regions. This is called diffraction. And we all know what the famous the famous wave particle duality experiment, which was proposed by Feynman many years ago and which has been repeated ample number of times in the laboratory, also helps us distinguish between wave particle duality. So if I have particles coming out of two slits and I have both slits open, I will get this curve P12. However, if I close one slit and keep the other one open, I will get either P1 and P2. And P12, if I keep both slits open, is just the additive sum of P1 and P2. This isn't the case for waves. If I close one slit, I would get I1 or I2. But if I keep both slits open, the pattern that I get is not the sum of the components. It's rather an interference pattern, which indicates that these particles are really strange. They're not particles per se, they are rather waves. And the counterintuitive thing with these objects is that you will get a wave-like interference pattern, even if you shot one particle, supposedly one particle into the system. So even one particle interferes, and it has nothing else to interfere with it, so it interferes with itself. This is the essence of the wave-particle duality. Now, does this have a role to play in quantum biological experiments? By the way, these are some experiments which on the right, you can see results of some real experiments that can do with photons. The other interesting idea in quantum mechanics, which is related to wave-particle duality, and then I'll ask the question, what does all of this have to do with biology, is taking multiple paths at the same time. Now, if I have a single electron or a single photon coming into an apparatus, suppose a photon is shining onto the lamina of a plant, a leaf of a plant, the blade of a leaf, and it's going to the photosynthetic center and the energy harvesting centers of the leaf. Now that single photon, how does that single photon reach the reaction center? Quantum mechanics tells us, if you're just dealing with a single photon, it tells us that if I have a source of light, S, and I have a detector, P, and if the detector sees a photon, which means that the photon has reflected off from some point on this mirror, then whatever the detector P sees is really a superposition of all of these paths. So the electron doesn't travel the photon doesn't travel in a straight line. Rather, it becomes a quantum field which permeates space, which is spread out in space, and it takes all of these multiple paths at the same time in a spooky coherence, which is called a quantum mechanical coherence. So what the observer at P observes is a superposition of these coherences. And it turns out that most of these paths destructively interfere, and only that path constructively interferes, which complies with the classical Snell's law. But nevertheless, the photon takes all of these paths at the same time. This is called quantum mechanics sum of histories approach. Even though an electron may move from point A to point B, rather, it doesn't move in a straight line. It rather becomes a quantum field, spreads out in space. All of those sub-components, sub-fields, they interfere with one another to give you one classically observed path. But really, there is a coherence. There is a superposition of histories. And that's what quantum mechanics predicts. Now, in photosynthesis, excuse me, just I need just a break, please.
Sabieh, I had one because our title is on avian navigation. Are you going to get onto an avian navigation? Okay.
Yeah, that's yes.
All right. Fine.
That's my last example. G. So in photosynthesis, we all know the photosynthetic process. Now, photosynthesis from a quantum mechanical perspective is really baffling, because the quantum efficiency of photosynthesis is almost 100%. If a photon reaches the light harvesting center in a leaf, and I'm not sure about the biological details there, that photon invariably is converted into two charges separated out by a certain distance. So this is a process that has 100% quantum yield. Now the idea is, if photosynthesis is such an important process, and it's such a process with such a high quantum yield, how can we explain it? The answer to this question actually is not really clear. There have been proposals that this is due to quantum interference. So if I have a photon coming in, it produces an exciton. An exciton is an excited state of an electron and a hole. And the classical view is that this electron wave reaches the reaction center in a hop-like fashion. The other possibility is that it rather goes as a quantum mechanical superposition, as a coherence that is spread out over the leaf, over all the chlorophyll centers, and eventually reaches the reaction center and gives almost 100% quantum yield. Now, this idea has not yet been verified beyond a shadow of doubt, but uh, this is work in progress. So the first idea that I talked about in my presentation was quantum mechanical tunneling. The second idea that I introduced is the role of superposition and coherences and whether it is important for photosynthetic processes. And the last example that I would like to give is about avian migration, the migration of birds. Now, this has also been a point of contention for many years. But now and there are different mechanisms of birds moving around the globe. But there is one process which is fundamentally quantum mechanical. And that lies in how electron pairs of free radicals, which comprise two electrons, how do they react to magnetic fields? We know that the Earth is a large magnet. It's a large terrestrial magnet, and there's a magnetic North Pole and a magnetic South Pole. So, and there's a geomagnetic field. Now, birds actually traverse this magnetic field, and the process is inherently quantum mechanical. By the way, there are other non-quantum mechanical descriptions of how different birds and different aquatic animals migrate, but that is beyond the scope of my discussion today. I would just skip this slide as well. So the molecule that plays the most important role in this process is the photosensitive cryptochrome. I don't know much about this molecule, so I would like to move on to the quantum mechanical part. So in the retina of the bird's eye, if we look at an inner structure, these are the two membranes, and this cryptochrome molecule is embedded and spatially restricted within this plane, which means that it does not have the ability to diffuse around or change its orientation much because it's spatially restricted. And this spatial restriction has an important aspect in the avian navigation process. And the basic idea is that when light, so cryptochrome is a photosensitive molecule. When a photon comes in, a single photon comes in, it produces two electrons, lone electrons. It produces a radical pair. And these electrons, as we know, are magnetic particles. An electron is a magnetic spin. Each electron behaves like a tiny magnet. And these tiny magnets are oriented with respect to one another. So they could be oriented parallel to one another, or they could be oriented anti-parallel to one another. And if we have parallel orientation of these electrons in a radical pair, this is generally called a quantum mechanical triplet state. And if these electrons are oriented in the anti-parallel form, this is generally called a singlet state. Now, this singlet and triplet state, this pair of electrons, they are coupled with one another, but they're also coupled with the nuclei in this system. This coupling is called the hyperfine interaction. On top of the hyperfine interaction, these electron pairs, the singlet and the triplet, also see the geomagnetic field, the Earth's magnetic field. And so now we have a system of two electrons. They are coupled with one another. They also interact with the nuclei through the hyperfine interaction, and they're also seeing a terrestrial magnetic field. And as they change their course on the Earth's surface, the terrestrial magnetic field is changing. So under the action of these changing magnetic fields, these singlets and triplets can interconvert. Now on the screen here, you would see in pictorial form what I've just described. Two electrons shown here in a triplet state. They are interacting with nuclear spins. So this is the electron nucleus system. Much of what happens in an NMR microwave experiment called an Andor experiment, and that's why you can simulate or perform these experiments in a simulated environment in the laboratory as well, and that's what many researchers do. This electron nuclear system is now placed inside a magnetic field. The orientation of that magnetic field is variable, dependent on the longitude and latitude of this bird on the Earth's surface. And the singlets, so here I also show a singlets pair, plus minus, spin up, spin down, and superposition will spin down and spin up. They are together with a triplet, a singlet of nuclear spins. So this entire system can then undergo an interconversion between the singlet and the triplet state. Whenever a spin is placed inside a magnetic field, if there is a relative orientation between the magnetic field and the spin is going to precess. It's going to go round and round and round and round. If we're talking about two spins, the singlets are continuously interconverting to the triplet state. The triplet state is coming back into the singlet state. So there's a precession in the singlet triplet plane. And dependent upon, that's why the singlet fraction or the amount of singlet in this electron nuclear system is going to be dependent on the position of this bird in the geomagnetic plane, and that is what determines what the bird actually sees. Now, we could also perform this entire calculation quantum mechanically. So we could start off with the Earth's field pointing on the north, east, and down direction, and then we can transform it to the avian frame of reference, to the frame of reference of the bird through a series of transformations. And then we could look at some initial singlet and triplet density matrix, which tells us what state the bird's electrons and nuclei started off with, and how do they evolve with time, given that the Hamiltonian for this process, which drives the process, is dependent on theta and phi, where theta and phi are the orientation or the location of the bird on the Earth's scale. So now all of this could be done numerically and analytically to a certain extent, and we could determine what the singlet and the triplet fraction is going to look like as the bird navigates its course on the Earth's surface. So one of my students did some of these calculations partially, and on the left and the right, you will see the fractions of the singlet and the triplet for different orientations of the bird. Now the bird can move in erratic ways. It can move in very interesting patterns on the Earth. It can change its latitude and longitude, but it can also scan its head up and down as it moves. And as it scans its head up and down as it moves, the angular negotiation of the magnetic field is also going to change. And that will lead to different fractions of singlet and triplet, and which eventually leads to different visual filters on what the bird actually views. So going from north to south, the view might look totally different if the bird went from south to north. So quantum mechanics is at play here, and this has been validated to considerable certainty. That
Sabieh, you have 5 more minutes.
I'm done. I can take questions. So I'm done, Shahid Sahab.
Okay, great. No, that's good. So maybe I'll ask the first question. आप अगर एक slide, can you go back one slide, because I didn't understand what results you आप present किए थे of your student. So I would like to understand that. Yeah, because I can't read the writing. वो ये so I can see that there's a panel of blue, 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 and then on the right there's a panel of red, red, red. But so what's happening there? आप जरा थोड़ा you can explain that, that would, I think, be helpful not only for me, but for others as well.
G. So if you allow, may I change the screen a little bit and show you another figure that
Yeah,
describes.
sure. Okay. Yeah.
All right. So just a minute, please. So basically what I've done here is that the retina, the bird's eye, is modeled as a hemisphere.
Okay.
Okay. So here I go. Let me share my screen again. Excuse me. Sorry, I'm slightly slow on this.
No, this is there. Yeah, you're coming up. Okay. Yeah. All right.
So this is a model of the retina.
Oops.
Sorry, it's excuse me. Sorry, it's just taking time. So the retina is modeled as a hemisphere.
Okay.
Hemisphere, there is a certain distribution of rods and cones. So there is a distribution of photosensitive molecules on this hemisphere. And this model can be taken from the biological model. Each point on this hemisphere is defined by two angles, theta and phi, which are the polar coordinates of that photosensitive molecule on the retina, right? Now the magnetic field could have a certain orientation. The orientation of the magnetic field is described by another set of direction cosines or angles alpha and beta. So now we have a pair of spins interacting with nuclei through the hyperfine interaction. We can set up a position dependent Hamiltonian. So each point of this hemisphere, which is demarcated by theta and phi, we will have a certain Hamiltonian. That Hamiltonian will also have the magnetic field coordinates inbuilt into it. So this Hamiltonian is going to propagate or change the quantum mechanical state of this electron nucleus system. And it's going to evolve with time. The singlets are going to go into the triplets are going to interconvert back into the singlets, and the entire system is also decohering with time. There is relaxation. You cannot keep, especially at room temperatures, at the temperature which the bird's eye is working, these coherences have to dissipate with time. So what we do is, in the figure, we let me move back to my original figure. Excuse me, sorry for this mess.
No, that's fine. I mean, I think we have a bit more, yeah. And then we also have a more extended Q&A. I just want to.
So if I move back to this figure here.
Yes.
On I don't know how to zoom in further. On the x-axis, we have the azimuthal angle phi.
Okay.
And on the vertical axis, we have the polar angle theta. So the hemisphere of the retina has been stretched and
I got you. Okay.
as a flat surface. Now on each point on the left panel, we are plotting in a color bar, in a color map, the singlet, the yield of singlets with time. At one point in time, we're averaging over a certain time period. So we're plotting the singlet yield for different values of the magnetic field. So if you go from top to bottom, the orientation of the magnetic field is changing. So which corresponds back to a varying longitude and latitude, the position of the bird on the Earth's surface. So as we go from top to bottom on these graphs, on these panels, we see that the bird sees a different filter function or a different visual intensity pattern as its longitude and latitude changes, right? So the singlet and the triplet yields in fact control this. It's a spin sensitive chemical reaction. The chemical reaction that proceeds after this process is spin dependent. If the singlet yield is higher, one kind of signal goes to the brain. and it registers differently than if the triplets yield were higher. So what we are seeing here are the singlet and the triplet yields. On the left, we have the singlet yields, and on the right, we have the triplet yields. And as we go from top to bottom, we have varying positions of the bird on the Earth's surface.
Great. Okay, no, because I've known about this problem, but it's good that you've actually worked it out and done simulations on it. So this is nice. I think
I think
I should shut up and let another quick question, because then Yunhua, anybody else has a couple of quick. Sriram.
Yeah, I just want to know, okay, I'd like to understand what it is about this problem that even at ambient temperatures or body temperature allows quantum coherence effects to somehow be pronounced here, whereas in other systems, in thermally similar situations, they are not. I've also, I appreciate that it does seem to be happening, but I'd like to understand why. What's different?
Generally, you see, the reason is clear here. This is very, I think I can answer your question. If you have a single, by the way, nuclear spins are long-lived as compared to electron spins, because the nuclear spins are they are inside the nucleus. They talk very little with the environment. That's why you can do NMR at room temperatures. Nuclear magnetic resonance, or MRI, works at room temperatures because the nuclear spins have a very long lifetime. That lifetime can be of the order of seconds, if not milliseconds. And that's why you can see sharp peaks in an NMR spectroscopy experiment. NMR is a high temperature experiment, but here we're dealing with electron spins coupled to nuclear spins. So if you just had a single electron spin, the coherence time of that electron spin would be of the order of microseconds to milliseconds, perhaps. Now, even at these high temperatures, if you produce a singlet state is a particular quantum mechanical state which spin up, spin down, but it's not just spin up and spin down, because that does not obey the Pauli exclusion principle. It's a superposition of spin up, spin down, minus spin down, spin up. So it's a special kind of antisymmetric state, which is odd under particle exchange. Now, this singlet state is a long-lived state because it commutes with the Hamiltonian. The Hamiltonian is really a magnetic field acting on spin 1 and a magnetic field acting on spin 2. Over the size of the molecule, the magnetic field is not going to change its orientation. So spin 1 sees the same magnetic field orientation as spin 2, which means that the Hamiltonian for this system is also isotropic for the two spins if they were in the singlet state. So the singlet state quantum mechanically lives in a decoherence free subspace because it's singlet. And that's why it's a long-lived state, and its coherence time can approach a second, even at room temperatures. By the way, the interconversion to the triplet state happens not because of the magnetic field, but because of the differential interaction of these spins with the nuclei, the differential hyperfine interaction. That's, if there were no hyperfine interaction, the singlet would never change into the triplet, and you would not see a visual pattern or a visual filter function, and the bird will not be able to tell whether it is going south or it's going north, it's going east, west. So the answer to your question that these are relatively longer lived states because they work in a decoherence free subspace, or they are singlet, and the singlet commutes with the magnetic fields Hamiltonian.
Okay, thank you. Thank you.
Okay, we should move on. Because there will be more extended question and answer, but I think Yunhua is already 5. Yeah, so Yunhua, I'll give you 5 minutes because we've kind of eaten a little bit into your talk. Yunhua, are you ready to go?